All right, good afternoon. This is the third of our three scheduled webinars. We've got two presenters today from DNR. Our first presenter is Steve Meyer. Steve's going to be discussing the freshwater resources of the Santee River Basin. Steve is DNR's freshwater fisheries regional coordinator for the coastal plain of South Carolina. He's been with the agency for 5 years and oversees sport fish population monitoring and helps guide management recommendations in that region. Our second presenter is going to be Dr. Joey Ballinger. He will be discussing aquatic saltwater resources in the Santee River Basin. Joey is the manager of the Inshore Fisheries Research Section at DNR's Marine Resources Research Institute, where he oversees the long-term monitoring efforts for estuarine finfish and coastal shark populations. He's been with DNR since 2009, managing life history studies or reef fish prior to late 2017 when he became the manager of the Inshore's fisheries section. Prior to his time at DNR, he was working on his PhD in biological oceanography, studying the population dynamic of head in the Chesapeake Bay at the Old Dominion University. Joey holds a master's in fisheries and allied aquacultures from Auburn University and has a BS in biology from Winthrop University. All right, so Steve, I'm going to turn it over to you. RBC members and others attending, if you have questions, I would generally encourage you just to save them until the end of each presentation. They'll both go about 20 or 25 minutes. But if you have something that you'd like to get clarification for, you might want to raise your hand or put it in the chat, and I may stop the presenter and ask for clarification along the way. But generally, let's try to hold our questions until each presenter is finished. So with that, I'm going to turn it over to Steve, and Steve, I'll let you run with it.
Hey, I'm Steve Meyer from the South Carolina Department of Natural Resources, and I'm the freshwater fish biologist on the coastal plain of South Carolina. My team and I monitor sport fish populations in our region and help make management recommendations. My presentation today is on the freshwater resources of the Santee River Basin. Today I'm talking about the fish and mussels in the lower Santee Basin, which includes the Congaree River, Lakes Marion and Moultrie, the Santee River, Cooper River, and Ashley River. There are 91 native fish species present in these drainages and 9 introduced species. Starting off at the upstream end of the system, we have Congaree National Park. The park boundary is 23 river miles downstream of Columbia and is home to 56 species of fish. The park protects 27,000 acres of floodplain geology and hydrology. It is the largest intact area of old-growth bottomland hardwood forest in the southeast. It sees major flood events, which cover 90% of the park and occur around 10 times a year. These floods are necessary to preserve the park's unique ecology and provide connectivity. Riverine floodplains support high levels of biodiversity and some of the most productive ecosystems on Earth. They're also extremely valuable economically in terms of the services they provide to society, including reduction of flood risk and support for highly productive fisheries. River flows through the park are highest during the late winter to early spring and typically lowest during late summer to early fall. And here's a picture of the floodplain at Congaree National Park. Now I'll talk about the sportfish species in the system. Sportfish, like the striped bass, are highly sought after by anglers. The landlocked reproducing population of striped bass in the Santee Cooper reservoirs was a unique phenomenon until recent discoveries in other reproducing populations around the southeast and out west. South Carolina was a pioneer in developing striped bass hatchery techniques as a direct result of the work at the Dennis Wildlife Center, which is here in Bono. Striped bass fisheries now exist in many reservoirs across the country. And you can see a fingerling striped bass grown in Bono on that picture on the right, little guy. Bass and crappie are also highly sought after in the system. We've got largemouth or Florida bass, black crappie, and white crappie, which are introduced They're popular for their fast growth rates in our system and trophy potential. We have several species of sunfish present in our system. The most popular from an angling perspective are bluegill, redbreast, and red ear or shellcracker. But here's some other species that are present as well, like flyer, pumpkin seed, spotted sunfish, and warmouth. Blue catfish are maybe the most fished for sport fish in our system. People travel great distances to fish for our trophy size Santee Cooper blue catfish. Flathead catfish can also get really big and capture the interest of fishermen. Channel cats don't get quite as big and are sought after for their excellent table fare. And several native catfishes are found in the drainage as well, including white catfish, brown bullhead, snail bullhead, yellow bullhead, and flat bullhead. Chain and redfin pickerel are also found throughout the system. Now I'm going to mention a few surveys that we take on to get a better grasp on the populations and trends of sport fish species in the system. One is standardized electrofishing targeting striped bass in the Congaree River. It took place over 5 days during spawning season between March 21st and May 1st last year. Visual determination of spawning condition indicated that most fish spawned by the 3rd week of April, and we had similar observations in the previous 3 years. Catch rates, measured in number of fish caught per hour of electrofishing, has decreased each year since 2021, which indicates that less adult striped bass are attempting to spawn and has guided stocking strategies and could potentially guide changes to the management regulations. Gill netting survey over the past 40 years has yielded a long-term data set on Lakes Marion and Moultrie. This gill net survey monitors many species, but specifically focuses on striped bass and blue catfish population trends in the lakes and looks at those, how they change over time. The data from this study helps guide management and stocking decisions in the lakes. And you can see on this pie chart the number and percent of sample for the top 5 species caught in last year's gillnet survey. Blue cats are typically the most abundant species and were again last year, with a whopping 452 blue catfish caught. Other common catches in descending order of abundance last year were gizzard shad, white perch, striped bass, long-nosed gar, and channel catfish. We do long-term electrofishing surveys on the lakes, alternating surveying each lake every other year. So last year was Lake Marion, and the year before that was Lake Moultrie. We're back on Lake Moultrie right now in 2025. Sites are randomly selected in each of 4 zones on the lake, and standardizing the amount of effort spent at each site has resulted in a long-term data set that can be used to observe trends in the population and, again, help guide those management decisions. And here are some pictures of some big bass we caught during the electrofishing survey, if there's any bass fishermen out there. We have a standardized sampling plan for freshwater portions of the South Carolina coastal rivers, targeting all sport fish. And we've completed surveys on each of the 3 coastal rivers in this basin, with our final year on the Ashley River taking place this upcoming summer. We'll be wrapping that up. Sport fish are not just important to the recreational anglers who obsess over them, they're also economically important. From the manufacturing of gear and boats, the sale of fuel for boats and transportation to and from the water, and the lodging and restaurants that the fishermen support, fishermen definitely aren't scared to spend lots of money on their favorite hobby. And while it's just a hobby for some, there are also fishing guides and commercial fishermen in the Santee system who rely directly on sport fish populations for their livelihood. This famous billboard you've probably seen as you cross Lake Marion on the I-95 bridge demonstrates just how important sport fish are culturally and to the local economy. Diadromous fish encompass both anadromous and catadromous spawning behaviors. Anadromous fish are those that live in the ocean and migrate up freshwater rivers to spawn. In the Santee system, this includes American shad, blueback herring, Atlantic sturgeon, and short-nosed sturgeon. Catadromous fish grow up in freshwater and migrate to the ocean to spawn. This includes American eels, which are present in the Santee system. We have DNR monitoring efforts for all of these species, but one of the coolest ones is acoustic tagging and tracking of sturgeon. So both Atlantic and short-nosed sturgeon get implanted with acoustic tags so that we can track their movement patterns as they pass receiver stations throughout the Santee system. The battery life on these tags is 10 years, so we can track individuals over a fairly long period. And it gives us a better idea of their movements throughout the seasons, so we can better protect these endangered species and their critical habitat. Atlantic sturgeon typically don't travel upstream of the dams below Lake Moultrie and Marion, but sometimes we see the short nose get upstream into the Congaree. A standardized sampling plan targeting juvenile American shad using boat electrofishing takes place June through October on the upper Santee and Congaree rivers. And this is to determine abundance, distribution, size, and outmigration timing of juvenile American shad. We use genetic analysis to determine the rate of natural reproduction versus hatchery contribution to the population. And while we're out there, we're also collecting a community sample, looking at the distribution and relative abundance of all fish species. Here are those fixed sites for the Juvenile American Shad Survey on the Congaree and Upper Santee. The Congaree sites are near US Highway 601 and Bates Landing. And the Upper Santee sites go from the confluence of the Wateree and Congaree rivers down to the outflow into Lake Marion. And here is the species breakdown of what all we caught in 2023. 10,278 total fish were captured, representing 51 species. American shad was the most abundant, followed by eastern silvery minnow, coastal shiner, whitefin shiner, bluegill, and red ear. Next, I'll highlight some of the Santee Basin fish species of greatest conservation need in the South Carolina State Wildlife Action Plan. The State Wildlife Action Plan, or SWAP, is a comprehensive plan that addresses the species that the state deemed had the greatest conservation need. Factors included in this decision are rarity, threats, lack of management funding, and lack of data. Starting off in the small swampy streams, you'll find some of these highlighted fish. You'll notice as we go through the next few slides, some are grayed out while others are highlighted. The highlighted fish are the main players in the stream that size we're talking about. And while the grayed out ones occur in the system, they might be found more abundantly in different size streams. So here in the small ones, we see Everglades pygmy sunfish, Carolina pygmy sunfish, American eels, banded sunfish, and swampfish. In the medium to large streams, you'll see these species of greatest conservation need. They can be commonly found in these habitats. Again, those other fish may be present here, but these are the ones that are the main players. Black banded sunfish, snail bullhead, flat bullhead, and American eel. And moving up to the big rivers, you'll find these species of greatest conservation need: striped bass, iron color shiner, white catfish, American eel, and robust red horse. Here is a species of greatest conservation need I'd like to go into more detail on. It's that robust red horse. Here's some pictures of these big, beautiful suckers. We sample these fish during their spawning season in their spawning habitat of rocky, gravelly shoals. Measure them, weigh them, and document location of capture. If we don't already have a pit tag in them, which is like a microchip in your pet, we give them one, which has a unique code for that fish, so we can recognize it if we catch it again, where we caught it before, and if we already have data on that fish. You can see the pit tag reader on the left. It is giving me a code for that fish that had been previously captured and tagged. We also take a genetic sample, which can tell us whether the fish genetics match the parents used in the hatchery, which would tell us whether it was wild or hatchery spawned. And then after all that, we release the fish. Robust red horse is a large and long-lived sucker species that was thought to be extinct for nearly 100 years. It was rediscovered in Georgia in 1991. And since then, Georgia, South Carolina, and North Carolina have worked together to recover the populations. These populations were found to be genetically unique in the Altamaha, Savannah, and Pee Dee rivers. And the discovery of pharyngeal teeth in the Santee Basin led researchers to believe that they were historically here as well. So Savannah strain fish were stocked in the Santee Basin to reestablish the population, and we're starting to see evidence of wild recruitment in the Santee Basin. But some of the main threats to this species are human development, which can degrade their spawning habitat. That human development comes in many forms, but construction of dams, reservoir development, and just overall developing in the watershed can cause that sedimentation to come in. They spawn over gravelly, rocky shoals, like I said, which can be silted in due to altered flow regimes and erosion. Now, often overlooked are freshwater mussels. They are soft-tissued invertebrates with no eyes. They are bivalves, meaning they have two halves, and they're filter feeders. And here are 11 freshwater mussel species found in the Santee Basin, which have made the swap list of species of greatest conservation need. Freshwater mussel species have the highest percentage of federally endangered species in North America, with 75 listed endangered species. Mussels provide several ecosystem services, the first being improving water quality by filter feeding. They filter feed on microorganisms, converting them into dissolved nutrients, which helps improve water quality. They are a food resource to many animals, providing a link from these microorganisms at the bottom of the food chain up to the predators like fish, waterfowl, turtles, and otters. And another ecosystem service they provide is creating fish and wildlife habitat improvement through burrowing, which also enhances the rate of nutrient release from the benthos. And you can see in that picture on the right, native freshwater mussels are being used in a chub mound. So that's where the chub suckers will lay their eggs. And here's some invertebrates using freshwater mussel shells as habitat. Mussels have this symbiotic relationship with fish. They provide all the aforementioned ecosystem services for the fish, and in return, fish provide mussels with a place for their larvae to grow and facilitate movement of their larvae around the system, since mussels themselves aren't very mobile. So during breeding season each year, freshwater mussels reproduce as males release sperm into the water column. So the sperm passively drift with the water currents and may enter females as they are siphoning and fertilize their eggs. The fertilized eggs develop into larvae called glaucidia. They resemble Pac-Man, like this one. And the glaucidia are microscopic in size and are held in the female's gills for future release into the water column. The glaucidia of freshwater mussels are parasitic and must attach to the gills or fins of a suitable fish host. Glaucidia usually die within a couple days after release from the female's gills if they don't attach to the right fish host. Some female mussels use specialized behaviors and structures, such as lures, in an attempt to release their glaucidia directly onto the fish. And if a glaucidium attaches to the proper fish host, it will remain on the fish for a couple weeks to a few months while it transforms into a juvenile mussel. And once mature, the juvenile mussel will drop off of the fish and settle into the substrate of the stream where it feeds and grows. If successful, the juvenile mussel will mature into an adult and the process will repeat itself. We've seen historic declines in mussel populations, all due to human impacts. So harvest, introduction of invasive species, and habitat degradation are all threats to mussel populations. In conclusion, I'd like to reiterate some of the freshwater resource concerns for the water plan. The importance of connectivity of ecologically functional floodplains through periodic flooding, spring flow requirements for spawning of endangered diadromous species and striped bass, year-round flow requirements for mussels and all life stages of fish to survive, and spawning habitat requirements of the robust red horse. Oops, I skipped a slide. I wanted to thank these people who are DNR staff that helped me by providing slides, pictures, and feedback on their respective areas of expertise. And with that, I'll take your questions.
Great, thank you, Steve. John posted a question in the chat earlier about what size of mesh do you use for gill netting?
So we use an experimental gill net, which means it has 7 different sizes of mesh, ranging from 2-inch up to 8-inch mesh stretched, so 1 to 4 bar. So 50 feet of each mesh size, all connected, and we have 1 floating net and 1 sinking net. So we're covering all fish sizes and all water columns as best we can.
And John, did you have a follow-up question if you want to unmute yourself?
I did, but I saw Sarah had a question up. So I guess I had a couple questions. I was curious. Do you have any indirect evidence that just having fish lifts at St. Stephen's and the lock at Jeffries, does that help mussels reproduce? I mean, does that help mussels migrate up into the lakes, as they attach to fish? I mean, it helps fish, but you have indirect evidence that it helps the mussels at all?
I'm not sure if we do have evidence of that. I would assume if fish are passing, that some number of those fish are passing with larval mussels attached to them, but I don't know if we have any real evidence of that. I would have to put you in contact with our mussel biologist. She might be able to answer your question better. Her name's Erica Basin, and maybe I could send you an email, or you could send me one after this. Your name is John.
Yeah, John Grego. Yeah.
Okay.
Yeah, I actually used to. I was with the consulting lab at the University of South Carolina, and we used to do a lot of consulting with the freshwater fisheries lab, particularly the one on Van Bookland Road in Eastover. So, yeah.
Sarah, you had a question or two. Do you want to unmute yourself and ask Steve your question?
Sure. So I thought the pie chart with all the fish, it looked like you recorded fish that were caught. Were these samples of fish that were caught, or were these fishermen that had to report the fish that they were caught? Like you had a whole bunch of different species on that pie chart.
Yeah, so anything in this presentation was fisheries independent data, which means it was a DNR sample. It's not based on recreational fishing. That would be what we were catching, either electrofishing or gill netting.
Okay. And Mike Wooten.
Thank you. I thought that was really exciting for people who like to fish. Yeah.
Thanks, Sarah. Mike Wooten had a question. It says, please briefly explain how electrofishing is accomplished.
Okay. So yeah, we do mostly boat electrofishing. There's also backpack electrofishing, which you can use in smaller streams. It's kind of a smaller scale version of the same thing where you're wading around with a backpack electroshocker in shallow water. But we use boats because we're in a lot of deeper water here in the coastal region. So our boat has a generator on it. It produces electric current, which travels out through the anodes, which drop over the front of the boat. And the boat acts as a cathode. So there's current going between those anodes back to the boat. And anything in that field sort of gets stunned. And we scoop it up with nets. We measure it, weigh it, throw it back, and we keep track of how much time we spent shocking. So our box that's administering the electricity has a timer of seconds spent shocking. So then if we catch 100 fish in an hour, we know relative abundance wise, we're catching 100 fish per hour in this stream. We can compare that to another stream and sort of standardize our effort that way.
Good. Okay. And Mike also had a question. He said, years ago, there was a relatively significant eel export business located on the Cooper, not far from the Tailrace Canal. Is that still in business? Do you know, Steve?
I have not heard anything about that. I'm relatively new to the area. I moved here 5 years ago when I got this job. I might be able to put you in contact with Bill Post, the diadromous fish coordinator, who might be able to answer that question better.
Okay.
So just to recap, I'm following up with John Gregory.
John Grego.
About mussels.
Correct.
And who had the eel question?
That was Mike Wooten, and I can send you both their email addresses.
Perfect. Thank you.
Mm-hmm. Any other
John,
questions? Yeah.
Oh, sorry, John. This is Michael Melchers. I was going to just say, I do believe there is still some export of glass eels. I don't remember if it's in the Santee or the Cooper rivers. Bill will definitely be able to confirm.
I can speak to that a little bit on your next presenter. There is a glass eel export. I believe it's around the Goose Creek Reservoir area in the Cooper River system. So,
thanks, Joey.
And John, did you have another comment or question?
Yeah, I was just kind of curious where these reports were generally available. I mean, I've found them invaluable in commenting on permits. Just having the information about sturgeon and mussels in particular. But are reports of your sampling efforts available?
Yes, we can share those with you. If you reach out to me or our fish chief, Rick or Ross Self, we can share these reports with you.
Okay, thanks.
And Steve, one of your earlier slides, you had you talked about the trend in striped bass, and I think it was striped bass that had been counted via electrofishing, but that trend was declining over the last 4 or 5 years. What do y'all? You said that's making you adjust your management efforts, but do you have any idea what to potentially attribute that decline to, or any ideas?
It's hard to say. There's a chance that it's just due to less fish in the population in general. There's also a chance that there's less fish trying to spawn wild. So that survey is on the Congaree River, which is the spawning grounds for striped bass in the Santee Cooper system. So there might be altered flows or weather happening, different climatic effects that might be causing these fish to decide, hey, we're not going to try this year. Or there might just be less fish in the system. So you're catching less on the spawning grounds. There might be less mature fish in the system. So there might be less trying to make that attempt. It's hard to say for sure, but it is something that we're monitoring and keeping an eye on, and it's helping guide our management decisions. So if we think that there's less fish in the lake, we might be stocking more stripers. If we think that we need to protect those spawning fish, maybe we will recommend that those spawning grounds be protected or those size categories of fish be protected.
Okay. The other question I had is, when we've heard from other DNR fisheries folks in the other river basins, I don't remember them explicitly mentioning electrofishing being done in some of the other reservoirs in the state. Is it unique to Moultrie and Marion that DNR does electrofishing, or do you do it in other reservoirs?
No, it's not unique. It's fairly common for all of the large public waters. We have a standardized bass electrofishing survey. And then depending on the system, which species are present and what the stakeholders are interested in might guide whether some lakes are looking at other species, including striped bass, catfish. And we have other ways of sampling those fish too. If not electrofishing, we're using nets, gill nets, trap nets. So yeah, most public waters do have some kind of sampling going on them, and you could talk to the regional coordinators in those areas more about the details there.
Okay. The other thing we've been doing in the other river basins is work with Dr. Brandon Peoples and Luke Bauer of USGS on the stream flow ecology relationships. Are you familiar at all with any of that work that's been going on?
I'm not.
Okay. I won't put you on the spot then and ask my next question. I may reach out to them, but they've been very helpful in tying decades worth of fisheries and macroinvertebrate data to flows in these wadable streams and rivers, so smaller streams, smaller rivers and streams. and showing kind of the relationship between different flow parameters, magnitude of flow, frequency of flow, timing of flow, and how that impacts species richness, abundance, diversity, that type of thing. It's been a useful exercise in the other basins, but down in the coastal area, like in the lower Savannah, Salcahatchee, it's been a little bit limited where we can do that just because of the type of rivers and streams. And I'm thinking in the Santee, the Congaree and the Cooper and Ashley are not what's considered wadable streams. So I'm not sure they're going to be able to do the type of relationship or develop the type of relationships that they've done in the past. But I may need to speak to them and see what they could do in this basin. So we'll come back to that later. Any other questions for Steve?
Hey, John.
Right. Yeah, one more. Go ahead, Scott.
Scott. Just so Steve knows, that work that John referenced done by Brandon Peoples and Luke Bowers, they used a lot of data from Mark Scott's team up at the Clemson regional office. So Mark Scott was involved in that. Just so you know, if you have any questions about it, you can probably reach out to him as well. Okay.
All right, will do. Thank you.
Yep.
Well, thank you, Steve. We're right at the bottom of the hour, so it's time to move on to Joey. Joey, if you want to take over the screen share and show your slides, we'll jump right into your presentation. And you're on mute still, Joey.
Good thing done, dude. So yeah, I was just trying to confirm that y'all could see my screen before I sort of jumped into this. So
we can. Yep, it looks good. We can see the full thing now.
All right, sounds good. Yeah, thanks for having me here today to quickly talk a little bit about some of the monitoring we do around coastal South Carolina and the estuarine and nearshore coastal environment. We'll go through a couple of things, but first of all, I just wanted to make you guys aware, if you're not aware, the South Carolina Department of Natural Resources, Marine Resources Division, has what we call a Marine Resources Research Institute, whose express purpose is to conduct research and run monitoring programs that can be used to assess the condition of our coastal marine resources, and hopefully provide the data required to address policy and management issues related to those resources. With this mission in mind, the Institute has 4 primary research areas: monitoring and assessing the condition of all major fishery species, monitoring the condition of other important biological resources and their associated habitats, conducting environmental studies to assess the effects of human activities, and also conducting some maricultural research designed to develop improved technologies for producing seafood or enhancing depleted fishery stocks through a stock enhancement program. With those research goals in mind, I'm going to share some brief information regarding the types of research scientists at their institute have conducted in the planning area. These include estuarine finfish research section efforts, particularly related to species monitoring through inshore fisheries, which is what I'm the manager of, as well as some of our stock enhancement and mariculture related activities. I'm giving one specific example on the impacts of tropical systems and how that relates to freshwater flows. I'll then briefly go into some oyster demographics project being conducted by our crustacean and molluscan research section. And then last but not least, I will end today talking about our South Carolina Estuarine and Coastal Assessment Program, or CCAP program, ran by an environmental research section. So jumping right into it, we'll start with species monitoring being conducted by inshore fisheries. So the Inshore Fisheries Group is a group tasked with conducting long-term monitoring and scientific research on the inshore marine fish species of South Carolina to better understand their populations and provide information useful for their sustainable management. Today, this is accomplished by a small team of biologists, with myself sort of leading this group since 2017. Inshore fisheries as a concept began in the mid-1980s with a primary goal being to monitor the abundance and population demographics of fish resources through a combination of monitoring programs, much like Steve talked about in the freshwater system, as well as collecting information from coastal anglers regarding the types, sizes, and ages they were capturing. At the same time that we began our monitoring programs, we also began a long-term tag recapture study on red drum and other miscellaneous species. Our research focuses on two primary species interest groups, estuarine finfish, such as spotted sea trout, southern flounder, sheepshead, black drum, and last but not definitely not least, red drum, as well as small and large coastal shark species such as Atlantic sharpnose, bonnethead, black-tip sharks, tiger sharks, etc. Over the years, this has been accomplished primarily by running different monitoring programs, starting initially with a rotenone and stopnet survey in the late 1980s and early 1990s, and continuing on through our contemporary programs today, which I'll talk a little bit more about, being our TramelNet survey, our coast band surveys, which use a variety of different gears, a contemporary adult red drum and shark longline survey, and a contemporary estuarine trawl survey. Since the late 1980s, the group has made over 54,500 collections across coastal South Carolina, averaging about 1,400 per year since 1986. At the same time, I mentioned that tagging program. Since 1986, we tagged somewhere greater than 144,000 individual fish across 45 different species, with sort of, I think it's the top 8 or 10 species being shown in the table, but by far the most being red drum. So those contemporary surveys, coastspan, electrofishing, TramelNet, estuarine trawl, and longline operate from Murrells Inlet in the north, as we added sampling there in 2024, all the way down to Calibogue Sound estuary in the south. I did want to point out, despite this broad coverage, we don't really have any specific long-term monitoring of marine fish resources within the Santee Delta region of the Santee River proper. However, I didn't really think about it. I didn't include a whole lot of discussion of that here. We do have quite a bit of monitoring in that Cooper River and Ashley River systems that, because of the diversion of water flows, I guess technically are sort of considered part of this lower Santee River basin. So if you want more information on that, I can pull that out later. Where we do have a lot of sampling around Santee is in Winyah Bay, just to its north, and Cape Romaine, just to its south. And at least with our marine resources, we see pretty strong synchrony and similar trends across the entire coastal South Carolina. So information from those areas can be analogous to what we could expect to occur in Santee River area. So our longest running survey, contemporary survey, is our TramelNet survey, which began as early as 1991 in some strata. The survey is designed to sample lower estuary salt marsh edge habitats used by a number of recreationally and ecologically important species. Today, this survey operates across all major estuaries of South Carolina, from Port Royal to Murrells Inlet in the north, including routine sampling of Cape Romaine since 1994 and Winyah Bay since 2003. Target species include those recreationally important species like red drum, sheepshead, spotted sea trout, and southern flounder. But just to point out, this survey has actually encountered 136 different species and over 500,000 organisms since 1991 across coastal South Carolina. Since 1991, just to give you a sense of how much effort we put into monitoring these coastal populations, if you lay each of our trammel nets end to end, the total distance spanned by those nets will be about 3,400 miles, which would put you somewhere near a small town called Tok, Alaska currently. You'll see several more of these figures, and each of these lines are basically, this was the distance after 5 years of sampling, 10 years, 15, and so forth. Our second longest running survey is our coastal shark survey, or what we generally refer to as our coastman survey. As it grew out of our attempts to understand how elasmobranchs, sharks and rays, use our coastal and estuarine waters as pupping grounds during the spring and summer. This seasonal survey operates in 3 general areas, St. Helena Sound, North Edisto River, and Northern Bulls Bay, with those Bulls Bay sites being the closest to the mouth of the Santee River. The survey targets small and large coastal shark species that regularly inhabit South Carolina estuarine waters by targeting salt marsh edge habitats adjacent to deep channels and high salinity areas. Our third survey, which Steve has already talked a little bit about as far as the similar gear, is our boat-based electric fishing survey, where we're using it to target a much different habitat than either the travel net or the coast span survey. It does generally operate over much the same spatial footprint of coastal South Carolina, but this survey is sampling upper estuary, brackish, still tidally freshwater river creek and riverbank habitats. It's in this habitat where, which is likely most affected by water quantity being discharged from inland fresh waters, at least from the marine perspective, as flows can lead to large shifts in salinity patterns experienced in these dynamic environments, with high flows leading to lower salinities and low flows leading to higher salinities. That is why in systems such as Winyah Bay in the north or the Combahee River in the south, which are particularly susceptible to flows, you have such a large swath or a broader area covered by the survey, because during dry years, you had to go further upriver to find those freshwater habitats. During low flow years, we had to go further downstream or vice versa. So that said, these habitats are a critical area for marine species, including red drum, southern flounder, and southern flounder. But it's also an area where we see substantial overlap in the distribution of marine and freshwater species. As the survey also encounters many freshwater species such as largemouth bass, brim, and numerous catfish species. And just to put it in perspective, since 2001, when this survey began, our inshore fisheries group has probably drifted somewhere on the order of 2,100 miles of South Carolina riverbanks, which will put us near Boise, Idaho. And this survey will be celebrating its 25th birthday in January of 2026, while our TravelNet survey is celebrating its 35th anniversary this year. Our fourth survey is our adult red drum and shark longline survey. Though it grew out of some early work in the 1990s, this survey was standardized in its current form in 2007 to collect more information across coastal South Carolina. While it targets its namesake species, this survey has encountered 61 different species and greater than 36,000 individuals along coastal South Carolina since 2007. And also, similarly, if you put we put a lot of hooks in the water with a survey, with over 250,000 hooks deployed since 2007, and those long lines will extend about 2,200 miles if laid end to end. Our last survey is our estuarine trawl survey, which is jointly run by my group, Inshore Fisheries, and the Crustacean and Molluscan Research Section of SCDNR. It's designed to collect information on numerous species across coastal South Carolina, just focusing on a different habitat, namely river channel habitats. But it also includes a range of invertebrates and finfish species, encountering 135 different species and nearly 1 million individuals since 2010. So regardless of the survey in general, we are using the different platforms to collect similar information. This includes, and we're doing this by bringing individuals on board the research vessel, identifying and measuring them, tagging and sacrificing a subset for tag recapture and life history studies, and then returning the majority back to the water at their collection location. At the same time, we collect information on water condition, including things like tidal stage, water temperature, salinity, and dissolved oxygen concentration. This allows us, regardless of the survey, to answer such questions as how has the overall abundance of the species changed through time? How has the size or age composition of individual species changed through time? Are there trends in any of those metrics? How do changes in environmental conditions affect the catch of individual species? And has there been any long-term trends in overall environmental conditions in this area surveyed? So while I can't go through all the different data products we produce herein, I did want to provide a couple of examples that might be useful to your committee. One of the types of data we can look at with any of the surveys is community structure and potentially relate that to environmental factors, such several of which may be related to water quantity. Luckily, each of these surveys are designed to sample specific habitat types, not necessarily specific species. Thus, they each tend to catch a wide range of species. For example, when we look at cumulative species richness, which is being shown here on the left axis, we observe the most diversity in the Charleston Harbor area, but one must realize we are sampling Charleston Harbor more intensely than we sample other estuaries in the state. In fact, we sampled Charleston Harbor about 3 times more frequently than we sampled Winyah Bay, and about 2 times more frequently than Cape Remain, respectively, since 2010. Keeping sampling intensity in mind, we actually get pretty comparable species richness from our more limited sampling in Winyah Bay and Cape Remain, with 81 and 82 of the 121 different individual species captured in our TravelNet surveys being caught there. Combined across both of those systems, 97 species have been captured since 2003, including 10 species not captured elsewhere in the state in this survey, those being banded drum, black crappie, black-nosed shark, blue runner, common carp, Gulf kingfish, largemouth bass, shrimp eel, smooth dogfish, and spot-fin killifish. I'll just note that these sort of represent a mix of marine species found in high salinity waters, such as those common in Cape Remain, as well as freshwater species likely picked up in Winyah Bay during high flow events. This is just a similar graph, but this time for our electrofishing survey, showing the number of species observed in our 5 different river systems sampled by this survey. We've observed 82 of the 116 species in Winyah Bay, which is the closest to the Santee River Delta. And this includes 4 species: American harvestfish, least killifish, and robust red horse, and striped killifish that we haven't seen elsewhere. As Steve mentioned, I was going to point out robust red horse as well, because it's very conservation priority species, a high conservation priority species. It's a candidate species for listing under the Federal Endangered Species Act, and it's historically reported to be found in the Santee River Basin. So why I'm bringing all this up is, while I haven't done it here, we could use such community level information in concert with environmental information to see how species assemblage changes with respect to environmental conditions, which could be useful when developing a river's waters plan. We can also use the data to look at questions concerning the timing of interactions with individual species to see if they are found seasonally or not, and if the timing differs by stratum. So that's what I'm going to do over the next few slides. I'm going to show the monthly catch per unit effort for 6 species commonly encountered by the TravelNet survey. And notice that the seasonal interaction of the survey with individual species varies depending on the species and estuaries in question. First of all, I'm going to start with blue crab, which shows a fairly consistent with higher catch rates in the summer, though catches per TravelNet tend to be higher in the less salty Winyah Bay area. Another species of high conservation concern are diamondback terrapins, which this survey interacts with. We clearly see that in both Cape Remain and Winyah Bay, we see a peak during the spring in both estuaries, though they do appear relatively more common in this case in Cape Remain. Red drum, probably the #1 sport fish, estuarine sport fish for the state of South Carolina, shows relatively equal catches throughout all months of the year, if with a slight peak, which is most pronounced in Cape Remain during July through September, with catches generally pretty equivalent between the 2 areas. That peak from July to September is due to the gear effect of basically young red drum recruiting to the gear when they're about 1 year old. Southern flounder, available to the gear from April through October, but not available during the colder months of the year and caught more frequently in Winyah Bay. And then last but not least, spot and striped mullet, which shows generally increasing catches throughout the year in both estuaries. I'm showing you this type of data because these seasonal patterns suggest that if freshwater inflows change substantially, only seasonally, or we have more extreme variability in water flows across the years, relative to what we currently see or have historically had, you likely will affect some species more than others. We can also look at annual trends in abundance of individual species to try to determine if their abundances are changing dramatically through time. That is what I'm showing here once again for those same 6 species for Cape Remain and Winyah Bay. Here, clearly the abundance of some species, represented by the black lines, are changing at different rates and at different times in the 2 estuaries. For some species, such as blue crab, red drum, southern flounder, and spot, it seems to be the changes in species abundance seems to be fairly similar during overlapping periods of time. If it's going up in one system, it's going up in both systems. If it's going down in one system, it's going down in both systems. You can see this in a similar decline of blue crab since the late 2010s, the general decline in red drum since the early 2000s, the general decline in southern flounder throughout the time series, and to the relatively stable to slightly increasing catch of spot in the most recent years. For others, there's less synchrony and changes in abundance between the 2 estuaries. For those 6 species I pulled out, that might be indicative like diamondback terrapin and striped mullet. Well, I'm only showing information here from 6 species from the Travelnet survey. We've had this type of information for about 75 different species from coastal South Carolina, mostly marine, but some freshwater when you're looking at data from all the different surveys. Because we have this information, if there are some specific questions regarding how populations respond to different environmental conditions, we may have the information needed to answer those questions. For example, if you're interested in salinity, which is related to freshwater volume in the estuary, may be an important driver of the abundance of individual marine species in estuarine environments, such as those found at the terminus of the Santee River Basin. We could use the abundance data available from individual surveys to investigate the relationship with salinity of individual species. It's what I'm showing here. Once again, focusing on the abundance of those 6 species used previously. What we see is that the catch of these species does vary with salinity, with some species being more prevalent at lower and intermediate salinities, and red drum and spot being more commonly at higher salinities in Winyah Bay and Cape Remain. So the reason I'm pointing this out is clearly these such relationships point to implications for estuarine species under changing freshwater flow regimes. For example, it might mean under lower flows, the salinity pattern of the estuary changes, resulting in higher salinities in a given area. While the fish may be able to move upstream to stay within their preferred habitat, if other components of the habitat are missing, this could still affect their viability. In contrast, for species that thrive at high salinities, such as spot, lower flows might be predicted to lead to increases in abundance, all else equal. Such relationships with salinity clearly would have some implication to water management. So what I'm really trying to give the group here is an appreciation of the types of data we have at SCDNR. Here I've just provided a snapshot of the types of questions that could be investigated using only a couple of examples. This isn't an exhaustive list, but if y'all have some specific questions, I'm happy to work with you guys later on or in future meetings to try to answer those questions. Next, I'll give a brief example of how research from our stock enhancement group has contributed to our understanding of red drum recruitment across coastal South Carolina. First, a brief history. SCDNR began a red drum stock enhancement program in the late 1980s. We were trying to, one of the primary reasons being due to the importance of red drum as a recreational saltwater game fish. As many are here are aware, climate change and increased weather variability has given rise to some new existential threats, including rising temperatures, sea level rise, et cetera, et cetera. As a tangible example of the impact, the graph here in the bottom left is showing the tropical activity in the Western Atlantic, suggesting it has increased since the late 1800s through 2020 and more recent years. With the increasing frequency of tropical activity along coastal South Carolina, we have seen increasing frequency of tropical-related flooding events in the late summer and early fall months, which can affect the recruitment success of estuarine species. This type of flooding, or lack thereof, can also be thought of as a proxy of what the effects of changing freshwater flows could be for estuarine-dependent species. And in this case, what we're showing here is the contribution of our stock enhancement program to our catches and our monitoring programs in either pre-storm events, which are in the light orange, or post-spawn. And the bottom line is what we're seeing is that in 2015, 2016, and 2018, when we have documented floods during the fall months, we see a greater contribution from post-flood stocking than pre-flood stocking, suggesting that flooding is leading to poor survival of natural recruits potentially. Now I'll just briefly talk about some research being done by our crustacean and molluscan research section, namely their oyster demographic project. One of the things we do here at MRI is to try to get a handle on distribution of oyster beds across coastal South Carolina. This layer is one of our layers that we produce, and it suggests there's somewhere in the neighborhood about 5,000 acres of oyster beds across the state. We are still missing data from some relatively large areas of the state, including, once again, unfortunately, around the mouth of the Santee River. But we know that the distribution of living oyster beds is not static through time. Thus, the Oyster Demographic Project began in 2015 to understand environmental effects on the growth and mortality of individual oyster beds across the state. As part of this, the project staff samples 35 or so stations annually, looking at the mortality. At each state, staff collect representative oysters, measure environmental conditions, and determine what percent of the oysters are living. They also collect length information. And what they generally have seen is since the project began during the 15-16 fill season, a general decline in the mortality rate or increased survivability. When we look at the average mortality across the state, all years and sites, we see that on average about 7.2% of oysters die annually. Though the single site in the Santee River Delta, which I believe is this one, shows the highest rate of mortality of all the sites sampled. The areas where we tend to see higher mortality tend to be those areas where we see either periods of flooding over the last few years, leading to estuaries turning completely fresh, or areas where we've seen more urbanization. And then really quickly, just finish up here with the environmental research section in the South Carolina Estuarine and Coastal Assessment Program. So SCDNR has monitored coastal conditions in regards to water quality, etc., through the CCAP program using broad indices that include water quality, nutrients, bacteria, and the health of the benthic community. These data are used to look for areas of degradation and for changes over time as the coast continues to develop. The newest report is available, summarizing findings from 1999 through 2022 coast-wide. It assesses stations based on a water quality index, a sediment quality index, and a biological condition index. Basically, each station determining whether it's poor, fair, or good for each of these metrics. Here is the all years combined water quality metrics for the central region, with the Santee River area being shown here in the top right. What's good is we're seeing a lot of green. We're not seeing a whole lot of red. Same thing, generally speaking, for sediment quality in the central region, although you are seeing more red sediment quality in the Santee River than you were for the water quality. And the last but not least is the biological condition. Once again, showing it for the central region, once again, seeing a sort of mixture of green, yellow, and red regarding the health of that benthic community. So as I said, these are all integrated together to develop an overall index of habitat quality. With this being the resultant habitat quality map for the central region using data from all sample stations from 1999 through 2022. For the central region, so you got Charleston here and Santee River Delta up here in the top right. Once again, most good as we're seeing mostly good to fair and very low poor, probably a decreasing level percentage of poor in recent years across the state. And with that, I'll just leave you with a couple of points of contact from our institute, and be happy to answer any questions.
Thank you, Joey. A lot of great information. Any questions from the RBC for Joey?
John, I have one. This is Jason from Charleston.
Go ahead, Jason.
So, I was in the Edisto River Basin Council, and, we've got a really unique patchwork of 8 basins across the state of South Carolina with, roughly half with very few dams in it, and the other half with a whole lot of dams and a lot, not a lot in between. I mean, you've got heavily regulated basins and you've got a few that are very unregulated. It seems like in regard to the water quality, the sediment, and the biological health, data that you gave, but also just talking about the potential damages to the ecosystem when you have greater variance in salinity and freshwater conditions, that you hear a lot of people talk about the downside of a regulated or dammed basin. Have you done any comparison between the regulated basins and the unregulated basins regarding the variability of some of these conditions? In other words, if you want a consistent ecosystem, are there not some pros, as long as there's adequate fish passage, to having more regular releases for more consistent salinity and freshwater environments?
Joey, you're on mute. Sorry.
Sorry, I'm not sure that we have looked at that in detail. I think that I can follow up and potentially see if Andrew Twill or Denise Sanger with their CCAP data has looked at that with any detail. As I said, I mentioned the Winyah Bay and the Combahee River system from a point of our electrofishing survey. The Combahee is an unregulated system, largely speaking, and we see wild natural swings and salinity profiles depending on flow rates. The Winyah is a little bit more regulated, and we still see some of those things. But yeah, I don't know if I have the answer to that question yet.
I do
Wish I
remember
did.
when we did the Edisto ecological presentations from the university, it was surprising to see in that unregulated basin how well the species adjusted to those natural swings, both within the low DO swampy portions of the river system and also in the coastal.
Any other questions for Joey? All right, we're just a little bit over our time, but thank you, Joey, for fitting in all that good information. Again, this is being recorded. We'll post this to the Santee River Basin Planning website. It should be up there maybe by the end of the week. And if anyone has other follow-up questions for either Steve or Joey, feel free to send those to me and I'll send them off and we'll try to get them addressed before the next RBC meeting. Our next RBC meeting is actually next Tuesday. We've got Scott Harder talking about actual demand projections. Ashley Pritchett is going to be doing a presentation on capacity use areas and groundwater management plans. We're going to have a discussion on drought management response strategies. And Brooke is going to be talking about groundwater, kind of the groundwater outlook. So we've got a full meeting. With that, again, thank you, Joey and Steve. I appreciate everything you did today, putting this all together. And meeting adjourned. Thanks, everyone.